So we've heard a little bit already about flaral diseases in the context of drugs. I'm going to be talking specifically about onchocerciasis or river blindness, as it's sometimes known, and particularly the TOVA initiative, the onchocerciasis vaccine for Africa. So I'm representing quite a large number of international collaborators. I'll come back to that point shortly. So we've already heard quite a bit about the disease and the life cycle, so I won't dwell on this, really just to emphasize that we've got these two main stages that Ivan referred to. The adults, which are in onchocerca volvulus, are found located within these nodules, which are subcutaneous, but these release many thousands of microfilariae, the first stage larvae. And it's the microfilariae that cause all of the disease in onchocerciasis. So they're responsible both for the intense itching and premature aging, depigmentation that you find in the skin, which incidentally is much more common than the effects on the eyes. But they also do cause ultimately the visual impairment when they die within the eye, and there's an inflammatory reaction to the Wolbachia endosymbionts we've heard about earlier as well. So in terms of disease burden, most of you will know there's been these series of global burden of disease studies recently that have been funded by the Bill and Melinda Gates Foundation. I think the latest one was published quite recently, so data from 2015. So there's still a lot of onchocerciasis out there. That's the first point I want to make, is that despite decades of control, it's estimated that there are nearly 16 million people infected in Africa with onchocerciasis. So incidentally, this is quite close to the disease prevalence estimates that were published by the World Health Organization for onchocerciasis in 1995. So they were obviously grossly underestimated back then. Skin disease, as I say, is very common, so most people with onchocerciasis have some form of skin disease of varying severity, causing over 1 million years lived with disability. With vision loss, we have about 1 million people affected and 80,000 years lived with disability. So control of onchocerciasis in Africa has this very long history, so actually it's well over 40 years of sort of formal top-down control programs, starting in 1974 with the onchocerciasis control program, which operated over in these countries here that are not highlighted in the extreme sort of western part of Africa. Incidentally, even before the 70s, there were local control programs that led to the local elimination in some cases, for instance, in Kenya. So the OCP closed back in 2002. It was superseded and extended dramatically across Africa in these countries, coloured in green by the African Programme for Onchocerciasis Control, which, as I alluded to earlier, is, of course, dependent almost entirely on mass distribution of ivermectin. So APOC was originally scheduled to run up to 2007, and then it had its mandate extended to 2015, so it's no longer in operation. There's a program called SPEN, which is now operating in Africa across several NTDs that are all with a focus on preventive chemotherapy, with a funding model which is more dependent on local government funding, so obviously some potential risk there for countries that are facing economic crises or conflicts. So some years ago now, WHO published some studies showing that after 15 to 17 years, ivermectin had eliminated onchocerciasis in some foci in Senegal and Mali, where there was seasonal transmission. They got very excited about this, as you might expect, and actually APOC became, before it stopped, it became an elimination program rather than a control program, and we can perhaps discuss later what all this terminology really means versus eradication as well. However, recent modelling suggests elimination with ivermectin alone could take until 2040, and that's even if various key problems with ivermectin are addressed. Some of these issues have been raised again earlier by people like Ivan from DNDi, potential of emerging resistance, particularly this really big problem with ivermectin, severe adverse events in some individuals that are heavily co-infected with loiasis, another flora worm. There's a failure to break transmission in many rainforest areas where we have perennial transmission rather than seasonal transmission. Of course, there's the sustainability and logistics of distribution over decades of this drug and getting it to areas where, for instance, Sudan's got a lot of onchocerciasis, but also a lot of problems with conflict. And of course, I wouldn't be here if we had an onchocerciasis vaccine. There is currently no vaccine for this disease. So that's really where we came in with TOVA. So again, the Onchocerciasis Vaccine for Africa initiative, which sort of started on both sides of the Atlantic to some extent, but I guess it had a bit of a longer history in the European side. So from the University of Edinburgh, David Taylor, and then myself coming in from Liverpool, primarily with European funding. So as you can imagine, we're not massive fans of Brexit, as I'm sure is the case for most people in the room. And then, again, through the EU, we had partners at the Natural History Museum in Paris working on the Listomysoides sigmodontis model, and also the same model at the University of Bonn. Bonn also worked, though, on human clinical trials, the human diseases. We have these links that were forged through European funding again in the endemic countries, in Kumasi in Ghana, and then in 2 sites in Cameroon, in Yaoundé and Boya. And then we brought in mathematical modelling with Imperial College London more recently, and also another group working on Listomysoides in Glasgow, Simon Babian's group. And then extending across to the NIH-funded partners in the US, we've got New York Blood Center, that's Sarah Lustigman, and her partners in Philadelphia, Baton Rouge, and in Houston at the Baylor College of Medicine. So, as I mentioned in the earlier panel discussion questions, that I do have some experience with, in a veterinary context, with this large animal model of onchocerciasis, Onchocerca richengi, which is a natural parasite of cattle and the closest relative of Onchocerca volvulus in humans. So these are nodules containing the adult worms, so quite similar to the human nodules. They're transmitted by the same species complex of black fly vectors, Samunium damnosum complex. These adult parasites, as I say, they form very similar nodules. So if you do histological sections of these worms and you stick these sections in front of an experienced pathologist and ask them which was from a cow, which is from a human, it's actually quite difficult to tell them apart. And the antigens from these worms are extremely similar, so you get a lot of cross-reactivity with the 2 species. So some years ago now, there was a vaccine field trial done in cattle with 8 recombinant antigens that had come out of an old program called the Ed McConnell-Clark program that ran through the sort of late 80s and into the 90s. And these 8 antigens in combination gave this modest but significant protection against patent infection. So in other words, the cattle did not these protected cattle did not develop microfilariae in the skin, which was very promising. As I mentioned, the microfilariae cause all of the disease, and they're also the transmissible stage of the disease. But the level of protection was perhaps not as high as we'd hoped. So since then, there's been a lot of work on really trying to pin down, do we really need 8 antigens? Are there other antigens out there that may be better than the original 8? So REL2 and CPI2 over here were both used in the original trial. The CPI that was used in that original trial was unmodified, and for various reasons to do with quite complex immunology we can discuss later if you wish, it was realized that unmodified form of the antigen was probably not a good vaccine candidate. So that might have actually reduced the efficacy in that original trial. This antigen that goes by the exciting name of 103, it's some sort of clone number or something, was not used in the original trial, but we think this is a very potent vaccine candidate. So depending on the model and the system we're using, we get a variety of different levels of protection, but in some of the systems where we can look at adults and mycofilariae sometimes together in the Littomosoides model, we have seen efficacy of greater than 50%, sometimes as high as 70%. And for the mycofilariae, we consistently get very high levels of protection of 80% plus. So using the Brugia malayi model in gerbils, this study was published last year by the Baton Rouge group, Louisiana, and they looked at REL2 on its own here, and 103 on its own, using just alum adjuvant, which of course is a licensed adjuvant, the main licensed adjuvant for humans. And they got protection of around about 40%, possibly higher, depending on exactly how you calculate protection. But what was really exciting was that if you used these proteins as a fusion or concurrently, so either physically linking the two molecules together or just giving both proteins separately but given to the same animal, the protection could go up to over 50%, and in this case we reached levels of about 60%. So probably if we were in the malaria game, everyone would have been jumping around the room very excited, but we know for river blindness the bar perhaps will be higher depending on what you believe, or perhaps not what you believe, since we heard from Roy Anderson earlier, what the data tells us about what level of protection we need to achieve for a vaccine that's targeting a worm pathogen. So I'm very pleased to be able to say that just recently Sarah Lustigman at New York Blood Center won quite a substantial NIH award to take 103 and REL2 in combination into our cattle model, which we use out in Cameroon with this natural infection. So these animals over the next few years will be receiving 103 and REL2 with alum, or with another adjuvant, a newer adjuvant called Advax2, which apparently has been through human clinical trials. And then the best of these, depending on which adjuvant works best, a new trial will be done to try and reproduce the original data to see if we get similar levels of protection. So the million-dollar question really has been alluded to earlier is around safety. And of course, helminths can generate allergic-type responses. They generate IgE responses. Some years ago, during the original hookworm vaccine trial in Brazil, phase 1b trial, that had to be stopped early because many volunteers got urticaria from a type 1 hypersensitivity reaction involving IgE on mast cells or basophils releasing histamine. So a key question for our vaccine, for those of you who remember this great film Marathon Man, is it safe? And we'll come back to that in the panel discussion, but I think our data, particularly on REL2 and 103, is very good, that actually we're confident they are safe, as safe as we can be before we get to phase 1. In terms of what else we need to do, many things shown on this chart, but thankfully we've got this funding in place for the cow model now. But really the big gap, and again I'd like to discuss this later, is this sort of classic gap that we've seen in the Valley of Death, how to get funding to take us through preclinical toxicity, GMP production, and the phase 1 trial itself. And we have our own target of trying to hit phase 1 trials by 2020. So I'd like to acknowledge the other TOVA coordinators, particularly David Taylor, who's actually retired from Edinburgh now and has an honorary position at Liverpool. Sarah Lustigman, who won that NIH funding at New York Blood Center. My colleagues in Cameroon, Vincent Tanya and Germanus Barr, who work on the cow model. Tom Klein, colleagues at LSU, Louisiana, for that data from the Brugia model. And of course, the funders, the European Union and the NIH. Thank you.